6. Van der Waals forces in drug design. The understanding of van der Waals interactions is critical in the field of drug design, especially for the development of small molecules that can bind to enzyme active sites or receptors. The affinity of a ligand for a target is often determined by its ability to form van der Waals interactions in addition to hydrogen bonds and electrostatic forces. Ligand binding. Drugs can be designed to interact specifically with the van der Waals surface of a receptor or enzyme, ensuring a high degree of selectivity. Molecular docking. In computational drug design, van der Waals interactions are an essential part of the algorithms used to predict how small molecules bind to their targets. Previous year questions PYQs with answers 1. CSIR Net Deck 2012. Which of the following interactions is most responsible for the stabilizing of protein tertiary structure? A. Ionic bonds. B. Hydrogen bonds. C. Van der Waals interactions. D. Covalent bonds. Answer. C. Van der Waals interactions. Van der Waals interactions, together with hydrophobic interactions, contribute to the packing and stability of protein tertiary structure. 2. CSIR Net June 2014. In a DNA molecule, which of the following interactions plays a role in stabilizing the double helix? A. Hydrophobic interactions between phosphate groups. B. Pi-pi stacking between adjacent purine and pyrimidine bases. C. Covalent bonds between sugar and phosphate groups. D. Ionic interactions between complementary base pairs. Answer. B. Pi-pi stacking between adjacent purine and pyrimidine bases. Pi-pi stacking interactions are van der Waals forces between the aromatic rings of adjacent nitrogenous bases in DNA. 3. CSIR Net Deck 2017. Which of the following statements regarding van der Waals forces is false? A. They are strong at very close distances. B. They are involved in molecular recognition. C. They increase in strength with the size of the molecules involved. D. They are electrostatic interactions between permanent dipoles. Answer. D. They are electrostatic interactions between permanent dipoles. Van der Waals forces include both temporary dipoles, London dispersion, and permanent dipoles, dipole-dipole interactions, but they are generally weak and short-range interactions. Let me know if you need further elaboration or the next lecture on enzyme mechanisms, bioenergetics, or any other topic from CSIR Net Life Sciences. You said, electrostatic, ChatGPT said. Here is your CSIR Net JRF Life Sciences high-quality lecture on Unit, Molecules and their interaction relevant to biology topic, electrostatic interactions. Electrostatic interactions, also known as ionic interactions, are one of the fundamental types of non-covalent forces in biological systems. These interactions occur due to the attraction between oppositely charged ions or the repulsion between like-charged ions. They are much stronger than van der Waals forces but weaker than covalent bonds. In biological molecules, electrostatic interactions are pivotal for maintaining structural integrity, facilitating molecular recognition, and enabling many biochemical processes such as enzyme catalysis, DNA-protein interactions, and signal transduction. 1. Nature of electrostatic interactions. Electrostatic interactions are the forces between charged entities, either atoms, ions, or groups of molecules, that result from their electric fields. The strength of these interactions depends on the magnitude of the charges and the distance between them. The Coulomb's law mathematically describes the force between two charges. F equals K dot operator divides Q1 dot operator Q2 divides R2 F equals R2. K dot operator divides Q1. Dot operator Q2 divides. Where F is the force between the charges. K is Coulomb's constant. Q1 and Q2 are the magnitudes of the two charges. R is the distance between the charges. This relationship indicates that electrostatic force is stronger at shorter distances and stronger with higher charges. 2. Role in protein structure and function. Electrostatic interactions are critical for the structure and stability of proteins. They are involved in the folding of proteins, their tertiary structure, and interactions with other molecules. These interactions often occur between charged side chains of amino acids, e.g., lysine, arginine, glutamic acid, and aspartic acid. Salt bridges, formed by the attraction between oppositely charged residues, e.g., between lysine and glutamate. These interactions can stabilize protein structures and help maintain active sites in enzymes. Protein-protein interactions. Electrostatic interactions are key for molecular recognition during enzyme-substrate binding or protein complex formation. They ensure high specificity and stability in protein complexes. 3. Electrostatic interactions in nucleic acids. In DNA and RNA, electrostatic interactions between the phosphate groups of the sugar phosphate backbone and positively charged ions, such as Mg squared, not help to stabilize the double helix and facilitate various processes such as DNA packaging, transcription, and replication. DNA packaging. The negatively charged phosphate groups of DNA interact with positively charged histone proteins, leading to the formation of nucleosomes in chromatin. DNA-protein interactions. Electrostatic interactions between charged residues in histones and DNA enable the binding of transcription factors to DNA during processes like gene expression regulation. Ionic conditions. The stability of the DNA helix is influenced by the ionic strength of the environment, where higher salt concentrations can stabilize the double helix by neutralizing repulsive charges on the backbone. 4. Electrostatic interactions in enzyme catalysis. In enzyme catalysis, electrostatic interactions are essential for the binding of substrates and the formation of transition states. These interactions contribute to the induced fit model of enzyme function, where the enzyme's active site undergoes conformational changes upon substrate binding. Enzyme substrate binding. Electrostatic interactions between the active site and the substrate can orient the substrate in a specific way that promotes catalysis. Charge stabilization. Enzymes may stabilize charge transition states during catalysis by interacting with charged residues in the active site. Cofactors. Many enzymes use metal ions, e.g., Mg squared, Zn squared, that coordinate with charged amino acid side chains, aiding in catalysis by stabilizing negative charges on substrates or transition states. 5. Electrostatic interactions in membrane biology. In cell membranes, electrostatic interactions are essential for the stabilization of lipid bilayers and the function of membrane proteins. Lipid bilayers. The hydrophilic phosphate heads of phospholipids interact electrostatically with water molecules, while hydrophobic tails face inward, forming a stable bilayer structure. Membrane proteins. Electrostatic interactions between membrane proteins and the lipid bilayer, as well as between different membrane proteins, regulate processes like signal transduction, ion transport, and cell communication. 6. Electrostatic interactions in drug design. In drug design, electrostatic complementarity is crucial for designing high-affinity drugs that interact specifically with their target molecules, whether enzymes, receptors, or other biomolecules. Ligand-receptor binding. The strength and specificity of ligand-receptor interactions are often governed by electrostatic forces between the ligand and the receptor's charged regions. Pharmacophore design. Electrostatic interactions are a key feature of the pharmacophore, the minimal structure required for a molecule to be active in the target receptor site. 7. Electrostatic interactions in molecular dynamics. In molecular dynamics simulations, the electrostatic potential of a system is essential for understanding the behavior of biomolecules. These simulations allow the prediction of how charged particles interact within biological systems, including protein folding, ligand binding, and protein-protein docking. Previous year questions PYQs with answers 1. CSIR Net Deck 2013. Which of the following interactions contributes to the stability of the alpha helix in proteins? A. Electrostatic interactions between side chains. B. Hydrophobic interactions between nonpolar side chains. C. Hydrogen bonding between the backbone atoms. D. Van der Waals interactions between side chains. Answer. C. Hydrogen bonding between the backbone atoms. Hydrogen bonds stabilize the alpha helix structure in proteins. 2. CSIR Net June 2015. In DNA, which of the following is true about electrostatic interactions? A. They contribute to the formation of the double helix by neutralizing charges on the DNA backbone. B. They are irrelevant to DNA structure. C. They help in the stabilization of base pairs. D. They are mainly involved in hydrogen bonding between the base pairs. Answer. A. They contribute to the formation of the double helix by neutralizing charges on the DNA backbone. The electrostatic interactions between the phosphate groups and positively charged ions help stabilize the double helix structure of DNA. 3. CSIR Net Deck 2018. The ionic interactions in proteins are primarily responsible for a. Peptide bond formation. B. Protein folding. C. Hydrogen bonding between peptide groups. D. Stabilizing the quaternary structure. Answer. B. Protein folding. Electrostatic interactions play a crucial role in stabilizing protein folding and contributing to tertiary structure. This concludes the lecture on electrostatic interactions. Let me know when you're ready for the next topic or need further clarification. You said, hydrogen bonding. ChatGPT said. Here is your CSIR Net JRF Life Sciences high-quality lecture on Unit, Molecules and their interaction relevant to biology topic, hydrogen bonding. Hydrogen bonding is a type of electrostatic interaction that occurs when a hydrogen atom covalently bonded to an electronegative atom, like oxygen, nitrogen, or fluorine, is in close proximity to another electronegative atom with a lone pair of electrons. This interaction is significantly weaker than a covalent bond but plays a critical role in the stability and function of many biomolecules. Hydrogen bonds are key players in various biological processes, including the structure of DNA, protein folding, enzyme-substrate interactions, and the solubility of compounds in water. The directionality and strength of hydrogen bonds make them highly effective in promoting molecular recognition and maintaining macromolecular stability. 1. Nature of hydrogen bonding. A hydrogen bond is formed when hydrogen is covalently bonded to a highly electronegative atom, commonly oxygen or nitrogen. This hydrogen is attracted to a lone pair of electrons on another electronegative atom. The strength of a hydrogen bond is typically in the range of 1 to 5 kcal per mole, much weaker than a covalent bond, approximately 50 to 100 kcal per mole. Despite their relative weakness, hydrogen bonds are highly significant in biological systems due to their directionality and specificity. There are two main types of hydrogen bonds. Intermolecular hydrogen bonds. These occur within the same molecule. Intermolecular hydrogen bonds. These occur between different molecules. 2. Role of hydrogen bonding in protein structure. In proteins, hydrogen bonds play a critical role in the formation and stabilization of their secondary and tertiary structures. These bonds are primarily responsible for maintaining the shape and function of proteins by stabilizing alpha helices, beta sheets, and protein folding. Alpha helix. Hydrogen bonds form between the carbonyl oxygen of one amino acid and the amide hydrogen of another amino acid 4 residues away in the polypeptide chain. This stabilizes the helical structure. Beta sheet. Hydrogen bonds form between backbone amide groups of different polypeptide strands, stabilizing the sheet-like structure. Tertiary structure. Hydrogen bonds contribute to the folding of the protein into its functional three-dimensional shape by stabilizing interactions between different parts of the protein. 3. Hydrogen bonding in nucleic acids. In DNA, hydrogen bonds are responsible for the specific pairing of base pairs. The base pairing follows Chargaff's rule, where adenine A pairs with thymine T through 2 hydrogen bonds. Cytosine C pairs with guanine G through 3 hydrogen bonds. The formation of these hydrogen bonds between complementary bases in 2 strands of DNA stabilizes the double helix structure. The hydrogen bonds ensure the specificity and accuracy of DNA replication and transcription processes. DNA replication. Hydrogen bonds between the base pairs are broken by enzymes during DNA replication, allowing the complementary strands to separate and new strands to be synthesized. RNA structure. In RNA, hydrogen bonds also help stabilize the secondary and tertiary structures of the molecule, particularly in the formation of stem loops and other structural motifs. 4. Hydrogen bonding in water. Water is often referred to as the solvent of life, due to its ability to form extensive hydrogen bonds. These interactions confer many of water's unique properties, such as its high boiling point, high specific heat, and high surface tension. These properties are crucial for biological systems. High specific heat. Water can absorb a lot of heat before its temperature rises, which helps maintain homeostasis in living organisms. Solubility of biomolecules. Water's hydrogen bonding enables it to dissolve many polar and ionic compounds, facilitating the transportation and interaction of biomolecules in cells and tissues. Surface tension. The hydrogen bonds between water molecules create surface tension, which is important in processes like capillary action and the formation of cellular structures. 5. Hydrogen bonding in enzyme-substrate interaction. Hydrogen bonds are key to enzyme-substrate binding. The active site of an enzyme forms hydrogen bonds with the substrate, which helps in the proper orientation of the substrate for catalysis. These interactions also contribute to the induced fit mechanism, where the enzyme undergoes a conformational change upon binding to the substrate, optimizing the catalytic process. Specificity. The enzyme's active site contains specific hydrogen bond donors and acceptors that match the functional groups of the substrate, ensuring high specificity. Stabilization of transition states. Hydrogen bonding can stabilize the transition state of a reaction, lowering the activation energy and enhancing the enzyme's catalytic efficiency. 6. Hydrogen bonding in drug design. In pharmacology, hydrogen bonding plays a crucial role in drug design, particularly in the binding of ligands to receptors or enzymes. Drugs are designed to form optimal hydrogen bonds with the target molecule, improving binding affinity and selectivity. Pharmacophore. The functional groups of a drug molecule that participate in hydrogen bonding with the target receptor form part of the pharmacophore. Hydrogen bond acceptors and donors. A drug molecule can be optimized by adjusting its structure to increase the number of potential hydrogen bond donors and acceptors, enhancing binding to the receptor. 7. Hydrogen bonding and other biological processes. Protein-ligand interactions. The binding of ligands to proteins, such as in signal transduction, often involves hydrogen bonds that stabilize the binding. Antibody-antigen interactions. The specificity of the antibody-antigen interaction is also influenced by hydrogen bonds, which contribute to the tight binding and recognition of antigens by antibodies. Cell signaling. Hydrogen bonding in molecules like G-protein coupled receptors, GPCRs, and kinesis helps propagate cellular signals. Previous year questions PYQs with answers 1. CSIR Net December 2015. Which of the following interactions is primarily responsible for the base pairing in DNA? A. Ionic interactions. B. Hydrogen bonds. C. Covalent bonds. D. Hydrophobic interactions. Answer. B. Hydrogen bonds. Hydrogen bonds between base pairs in the DNA double helix stabilize the structure and contribute to specificity during replication. 2. CSIR Net June 2017. Hydrogen bonds are important in stabilizing the secondary structure of proteins. Which of the following is true regarding the role of hydrogen bonds in proteins? A. They form between the side chains of amino acids. B. They stabilize the alpha helix and beta sheet structures. C. They contribute to the formation of peptide bonds. D. They hold the tertiary structure of proteins intact. Answer. B. They stabilize the alpha helix and beta sheet structures. Hydrogen bonds form between the backbone amide groups in proteins, stabilizing the alpha helix and beta sheet secondary structures. 3. CSIR Net Deck 2018. The specificity of the double helix formation of DNA is mainly due to a. Hydrophobic interactions between the base pairs. B. Hydrogen bonding between complementary bases. C. Covalent bonds between the sugar phosphate backbones. D. Electrostatic interactions between the strands. Answer. B. Hydrogen bonding between complementary bases. Hydrogen bonds between adenine, thymine, and guanine cytosine base pairs ensure the specificity and stability of the DNA double helix. This concludes the lecture on hydrogen bonding. Let me know when you're ready for the next topic or need further clarification. You said, hydrophobic interaction. ChatGPT said. Here is your CSIR Net JRF Life Sciences high quality lecture on Unit, Molecules and their interaction relevant to biology topic, hydrophobic interactions. Hydrophobic interactions refer to the tendency of nonpolar molecules or parts of molecules to aggregate in aqueous solutions, away from the water molecules. This phenomenon occurs due to the inability of nonpolar substances to form favorable interactions with water molecules, which are highly polar. While not a traditional chemical bond, hydrophobic interactions play a crucial role in shaping the structure and function of many biological macromolecules, including proteins, lipids, and nucleic acids. 1. Nature of hydrophobic interactions. Hydrophobic interactions arise from the unfavorable enthalpy change that occurs when nonpolar molecules are exposed to water. Water molecules surround hydrophobic nonpolar molecules and disrupt the hydrogen bonding network that exists in bulk water, leading to a decrease in the system's entropy. To minimize this energetic cost, nonpolar molecules tend to aggregate together, reducing their surface area exposed to water and maximizing the water molecule's hydrogen bonding network. This aggregation leads to an increase in the entropy of the surrounding water, making the system more thermodynamically stable. Unlike covalent or ionic interactions, hydrophobic interactions are entropic in nature and are driven by the increase in disorder in the surrounding solvent, rather than direct attractive forces between the molecules themselves. 2. Hydrophobic effect. Hydrophobic interactions refer to the tendency of nonpolar molecules or parts of molecules to aggregate in aqueous solutions, away from the water molecules. This phenomenon occurs due to the inability of nonpolar substances to form favorable interactions with water molecules, which are highly polar. While not a traditional chemical bond, hydrophobic interactions play a crucial role in shaping the structure and function of many biological macromolecules, including proteins, lipids, and nucleic acids. 1. Nature of hydrophobic interactions. Hydrophobic interactions arise from the unfavorable enthalpy change that occurs when nonpolar molecules are exposed to water. Water molecules surround hydrophobic nonpolar molecules and disrupt the hydrogen bonding network that exists in bulk water, leading to a decrease in the system's entropy. To minimize this energetic cost, nonpolar molecules tend to aggregate together, reducing their surface area exposed to water and maximizing the water molecule's hydrogen bonding network. This aggregation leads to an increase in the entropy of the surrounding water, making the system more thermodynamically stable. Unlike covalent or ionic interactions, hydrophobic interactions are entropic in nature and are driven by the increase in disorder in the surrounding solvent, rather than direct attractive forces between the molecules themselves. 2. Hydrophobic effect in protein folding. The hydrophobic effect plays a central role in the folding of proteins into their native three-dimensional structures. Nonpolar amino acid residues, such as valine, leucine, and phenylalanine tend to cluster in the interior of the protein, away from the aqueous environment. This inward folding of hydrophobic residues is driven by the need to minimize exposure to water and is crucial for the formation of a stable, functional protein. Protein folding. The hydrophobic effect contributes to the formation of tertiary structure by driving the protein to fold in a way that exposes polar residues to the solvent and buries nonpolar residues in the interior. Protein misfolding. If hydrophobic residues are exposed to the aqueous environment, this can result in misfolding and aggregation, contributing to diseases such as Alzheimer's disease and Parkinson's disease. 3. Hydrophobic interactions in membrane formation. In biological membranes, the hydrophobic effect is responsible for the formation of lipid bilayers, which are the fundamental structure of cell membranes. The hydrophobic tails of phospholipids face each other in the interior of the membrane, while the hydrophilic heads are oriented towards the aqueous environment on both sides of the membrane. Lipid bilayers. The hydrophobic interactions between the nonpolar fatty acid tails drive the self-assembly of the lipid bilayer structure, which acts as a selective barrier to the movement of ions and molecules. Membrane proteins. Membrane proteins that span the lipid bilayer typically have hydrophobic regions that interact with the lipid tails and hydrophilic regions that interact with the aqueous environment inside and outside the cell. The integrity of the cell membrane is thus largely maintained by hydrophobic interactions, allowing cells to compartmentalize their internal environments and regulate substance exchange. 4. Hydrophobic interactions in nucleic acids. Hydrophobic interactions also play a role in the stabilization of nucleic acid structures. In the double helix of DNA, the hydrophobic bases are stacked inside the helix, away from the aqueous environment. This base stacking interaction, driven by hydrophobic forces, contributes to the overall stability of the DNA molecule. Base stacking in DNA. The hydrophobic effect between the aromatic rings of the purine and pyrimidine bases in DNA stabilizes the double helix by creating attractive forces that contribute to the overall helical structure. RNA structure. Similarly, hydrophobic interactions play a role in the secondary and tertiary structure of RNA, where nonpolar residues are buried within the molecule to avoid contact with water. 5. Hydrophobic interactions in ligand binding. Hydrophobic interactions are essential in ligand binding to receptors or enzymes. Many drug molecules and natural ligands bind to their targets through hydrophobic interactions, where the nonpolar regions of the ligand interact with nonpolar areas of the receptor or enzyme. Receptor ligand binding. The binding of nonpolar ligands to hydrophobic pockets in the receptor protein is often facilitated by hydrophobic interactions. This is particularly important in processes such as signal transduction and enzyme catalysis. Pharmacology. Drug molecules that are hydrophobic often have a higher affinity for their targets because they can effectively interact with hydrophobic sites on the receptor or enzyme. This characteristic is key in the development of many hydrophobic drug molecules. 6. Hydrophobic interactions in protein-protein interactions. Hydrophobic interactions also play a critical role in protein-protein interactions. When proteins interact with each other, hydrophobic residues often cluster together in the interface between the proteins, while polar residues are pushed to the exterior. Protein complex formation. Hydrophobic interactions contribute to the specificity and affinity between interacting proteins, and this is often observed in processes like protein-protein docking or enzyme substrate binding. Membrane protein complexes. In membrane proteins, hydrophobic interactions between proteins and lipids help maintain the stability and functionality of membrane-bound protein complexes. 7. Hydrophobic interactions in drug design. In drug design, understanding hydrophobic interactions is crucial for designing molecules with optimal binding properties. Drugs are often engineered to fit into hydrophobic pockets of target proteins, maximizing the hydrophobic surface area exposed to the ligand. Molecular docking. The success of many drugs depends on the ability of the molecule to interact with hydrophobic residues in the active site of the target enzyme or receptor. Thus, the hydrophobic effect is key in developing effective therapeutic agents. Hydrophobicity and bioavailability. Hydrophobic drugs are often designed to be lipophilic, allowing them to pass through cell membranes and reach their target sites effectively. 8. Hydrophobic interactions in biological systems. Protein aggregation and diseases. The failure of hydrophobic interactions in protein folding and stability can lead to protein aggregation, which is a hallmark of many neurodegenerative diseases like Huntington's disease, Alzheimer's disease, and Parkinson's disease. Chaperones. Molecular chaperones help refold misfolded proteins, preventing unwanted hydrophobic interactions that could lead to aggregation. Previous year questions PYQs with answers 1. CSIR Net December 2015. Which of the following interactions is primarily responsible for the stabilization of the protein's tertiary structure? A. Hydrogen bonds. B. Ionic bonds. C. Hydrophobic interactions. D. Disulfide bonds. Answer. C. Hydrophobic interactions. Hydrophobic interactions between nonpolar residues help stabilize the tertiary structure by driving them into the interior of the protein. 2. CSIR Net June 2017. Which of the following biomolecular interactions is responsible for the self-assembly of the lipid bilayer in biological membranes? A. Hydrophilic interactions. B. Hydrophobic interactions. C. Ionic bonds. D. Van der Waals interactions. Answer. B. Hydrophobic interactions. The hydrophobic effect causes the nonpolar lipid tails to aggregate together, forming the core of the lipid bilayer. 3. CSIR Net Deck 2018. Hydrophobic interactions in proteins primarily contribute to which of the following? A. Primary structure. B. Secondary structure. C. Tertiary structure. D. Quaternary structure. Answer. C. Tertiary structure. Hydrophobic interactions are primarily responsible for the folding and stabilization of the protein's tertiary structure. This concludes the lecture on hydrophobic interactions. Let me know when you're ready for the next topic or need further clarification. You said. Principles of Biophysical Chemistry. ChatGPT said. Here is your CSIR Net JRF Life Sciences High Quality Lecture on Unit, Principles of Biophysical Chemistry. Introduction to Biophysical Chemistry. Biophysical chemistry is the branch of chemistry that applies the principles of physics and chemistry to understand the structure, function, and interaction of biological macromolecules, such as proteins, nucleic acids, lipids, and carbohydrates. This field bridges the gap between biology and chemistry by providing tools and techniques to study the molecular properties and behaviors of biological molecules. Biophysical chemistry focuses on understanding the thermodynamics, kinetics, and structural characteristics of biological systems, as well as the interactions that dictate molecular behavior. Techniques used in biophysical chemistry include spectroscopy, calorimetry, and chromatography, among others, which help characterize molecular interactions, determine the structure of biomolecules, and study their behavior in various environments. 1. Thermodynamics in biophysical chemistry. Thermodynamics is fundamental to understanding the behavior of biological systems. In biophysical chemistry, thermodynamics describes the flow of energy and the changes in the entropy and enthalpy of a system. The primary concepts include free energy delta G. The Gibbs free energy change delta G determines the spontaneity of a process. In biochemical reactions, a negative delta G indicates that the process is thermodynamically favorable and can proceed spontaneously, while a positive delta G implies that the process requires energy input. Delta G equals delta H minus T delta S. Delta G equals delta H minus T delta S where delta H equals change in enthalpy heat content. T equals temperature in Kelvin. Delta S equals change in entropy degree of disorder. Enthalpy delta H the total heat content of the system, which indicates whether a reaction is exothermic releases heat or endothermic absorbs heat. Entropy delta S the measure of the disorder or randomness in the system. A positive change in entropy favors the spontaneity of a process. Equilibrium constant K. The equilibrium constant K describes the balance between reactants and products in a reversible reaction, and is related to the free energy change by the equation delta G equals minus RT ln K. Delta G equals minus RT ln K where R equals universal gas constant. T equals temperature in Kelvin. K K equals equilibrium constant. 2. Kinetics in biophysical chemistry. Kinetics in biophysical chemistry studies the rates of biochemical reactions and the factors that influence these rates. It involves the measurement of reaction velocity and understanding the mechanisms by which reactions occur. Reaction rate V. The rate of a reaction can be defined as the change in concentration of reactants or products over time. It is often expressed in terms of the rate constant K in the equation. V equals KAN versus equals KAN, where V equals reaction rate, K equals rate constant, A equals concentration of reactant, N equals order of reaction. Michaelis-Menten kinetics. In enzymatic reactions, the reaction rate is influenced by the concentration of the enzyme and substrate. The Michaelis-Menten equation describes the kinetics of many enzymes. V equals V max S Km plus S versus equals Km plus S V max S, where V max equals maximum reaction velocity. S equals substrate concentration. Km equals Michaelis constant, substrate concentration at half V. Max V max. Activation energy AIA. The activation energy is the energy required to initiate a chemical reaction. In biophysical chemistry, activation energy is important in understanding the efficiency of enzymes and catalysts in lowering the energy barrier for biochemical reactions. 3. Spectroscopic techniques. Spectroscopic techniques are used to study the structure and dynamics of biological molecules. These methods involve the interaction of electromagnetic radiation with matter, providing information about the molecular composition, structure, and dynamics. UV visible spectroscopy. This technique measures the absorbance of ultraviolet or visible light by molecules, typically used to study the absorption spectra of nucleic acids and proteins. It can provide information about the concentration of molecules in solution. Circular dichroism (CD). Circular dichroism spectroscopy is used. Circular dichroism (CD). Circular dichroism spectroscopy is used to study the secondary structure of proteins and nucleic acids. It measures the difference in the absorption of left and right circularly polarized light by chiral molecules, allowing for the determination of alpha helical and beta sheet content in proteins. Fluorescence spectroscopy. This technique measures the emission of light by molecule after it absorbs light at a specific wavelength. It is widely used to study protein folding, ligand binding, and molecular interactions by labeling specific parts of molecules with fluorescent probes. Nuclear magnetic resonance (NMR) spectroscopy is used to determine the three-dimensional structure of proteins, nucleic acids, and other biomolecules in solution. It provides detailed information about the chemical environment of atoms in a molecule. X-ray crystallography. This is the most powerful technique for determining the atomic level structure of proteins and nucleic acids. X-ray diffraction patterns are analyzed to create high-resolution 3D models of macromolecules. 4. Calorimetry. Calorimetry is the measurement of heat changes in biological processes, particularly useful in studying the thermodynamics of molecular interactions. Differential scanning calorimetry (DSC). DSC measures the heat required to increase the temperature of a sample and is used to study protein unfolding and ligand binding. It can provide information about melting temperatures, enthalpic changes, and enthalpy entropy compensation in biomolecular processes. Isothermal titration calorimetry (ITC). ITC measures the heat change during the binding of two molecules, providing detailed information on binding affinity, stoichiometry, and thermodynamic parameters such as enthalpy and entropy changes in molecular interactions. 5. Chromatographic techniques. Chromatography is a technique used to separate and analyze different components of a mixture, including proteins, lipids, nucleic acids, and small molecules. Common chromatographic methods include high-performance liquid chromatography (HPLC). HPLC is used to separate, identify, and quantify biomolecules in complex mixtures. It is commonly used for protein purification, analysis of small molecules, and separation of nucleic acid fragments. Gas chromatography (GC). This method is used to analyze volatile compounds. It is often coupled with mass spectrometry (MS) for detailed analysis. Affinity chromatography. This technique uses specific ligand receptor interactions to purify biomolecules, such as proteins or nucleic acids, based on their affinity for a specific ligand. 6. Molecular modeling and computational methods. Computational techniques in molecular modeling allow the prediction and visualization of the structure and dynamics of biological molecules. Techniques such as molecular dynamics simulations help understand protein folding, ligand binding, and the conformational changes in biomolecules over time. Previous year questions (PYQs) with answers 1. CSIR Net December 2015. Which of the following spectroscopic techniques is most suitable for determining the secondary structure of proteins? A. UV visible spectroscopy. B. Nuclear magnetic resonance (NMR). C. Circular dichroism (CDD). Fluorescence spectroscopy. Answer. C. Circular dichroism (CD). CD spectroscopy provides information about secondary structure elements such as alpha helices and beta sheets in proteins. 2. CSIR Net June 2017. The activation energy of a biochemical reaction is the energy required to initiate the reaction. B. Form the intermediate complex. C. Achieve the equilibrium state. D. Break the bond between reactants. Answer. Initiate the reaction. Activation energy is the minimum energy required for the reactants to undergo a chemical transformation. 3. CSIR Net Deck 2018. Which of the following techniques is primarily used to determine the three-dimensional structure of proteins? A. Fluorescence spectroscopy. B. X-ray crystallography. C. High-performance liquid chromatography. HPLC. D. Mass spectrometry. Answer. B. X-ray crystallography. X-ray crystallography is the gold standard for determining the high-resolution three-dimensional structures of proteins. This concludes the lecture on principles of biophysical chemistry. Let me know when you're ready for the next topic or need further clarification. You said, PH. ChatGPT said. Here is your CSIR Net JRF Life Sciences high-quality lecture on unit, pH, buffers, and biological significance. Introduction to pH. The term pH refers to the potential of hydrogen and is a measure of the acidity or alkalinity of a solution. It is calculated as the negative logarithm of the hydrogen ion concentration. H+. In a solution, pH equals minus log H+. pH equals minus log H+. A pH of 7 is considered neutral, indicating equal concentrations of hydrogen ions. H+, and hydroxide ions. OH-, O-. A pH lower than 7 indicates an acidic solution more. H+, ions than OH-, O-. A pH greater than 7 indicates an alkaline or basic solution more. OH-, O-. Ions than H+. For biological systems, pH is an essential factor in determining the structural and functional properties of biomolecules, such as proteins and enzymes, which are often pH sensitive. The role of pH in biological systems. In biological systems, the pH plays a critical role in maintaining the structure and function of biomolecules, ensuring proper enzyme activity and metabolic function. Protein structure. The tertiary and quaternary structures of proteins are highly dependent on ionic interactions and hydrogen bonds, both of which are influenced by pH. For example, changes in pH can disrupt ionic bonds, leading to protein denaturation or alteration of the active site, affecting enzymatic activity. Enzyme activity. Enzymes exhibit optimal activity at specific pH values. For example, pepsin, an enzyme found in the stomach, functions best at a highly acidic pH of around 2, while alkaline phosphatase is most active in basic conditions around pH 9 to 10. Metabolic pathways. Many cellular metabolic pathways involve pH-sensitive reactions. For example, glycolysis in the citric acid cycle can be disrupted if the pH deviates significantly from the physiological range, approximately 7.4. Ionization of biomolecules. The ionization state of biomolecules, e.g., amino acids, nucleotides, is influenced by pH. The ionization of functional groups in amino acids, such as carboxyl groups and amine groups, affects their charge and, consequently, the protein folding and interaction properties. Buffers and their biological significance. A buffer is a solution that resists changes in pH when an acid or base is added. Buffers play a crucial role in maintaining pH stability in biological systems, ensuring that cellular processes occur within an optimal pH range. Buffer capacity. The ability of a buffer to resist changes in pH depends on the concentration of the buffer components and the pKa, acid dissociation constant, of the acid-base pair used. Henderson-Hasselbalch equation. The relationship between the pH of a buffer solution, the pKa of the acid, and the concentration of the acid, HA, Ha and its conjugate base, A minus A minus, is given by the Henderson-Hasselbalch equation. pH equals pKa plus log A minus HA. pH equals pKa plus log Ha, A minus, where pKa is the pH at which the acid is 50% dissociated. A minus A minus is the concentration of the conjugate base, the ionized form. HA. Ha is the concentration of the acid, the undissociated form. This equation helps predict the pH of a solution based on the concentrations of the acid and its conjugate base. Biological buffers. In living organisms, several buffer systems help maintain pH within a narrow range, critical for optimal cellular function. The most important biological buffers include bicarbonate buffer system. This is the primary buffering system in human blood and extracellular fluid. The system consists of carbonic acid HCO and bicarbonate ions HCO. The bicarbonate buffer operates in the lungs and kidneys to maintain the blood pH at around 7.4. Reaction. CO gas plus. HO rightwards harpoon over leftwards harpoon. HCO rightwards harpoon over leftwards harpoon. H plus plus. HCO minus. CO gas plus HO rightwards harpoon over leftwards harpoon HCO H plus. Plus HCO minus. The equilibrium between carbon dioxide, carbonic acid, and bicarbonate helps buffer the pH in the blood. Phosphate buffer system. The phosphate buffer system operates inside cells and in the kidneys. It is similar to the bicarbonate buffer but involves phosphoric acid HPO and its conjugate base HPO and HPO squared. The phosphate buffer is effective in the intracellular environment, where the concentration of phosphates is relatively high. Protein buffering. Proteins can also act as buffers due to the presence of amino groups NH and carboxyl groups COOH, which can gain or lose protons depending on the pH. Histidine residues in proteins are especially significant for buffering because they have a pKa near physiological pH around 7.4. Hemoglobin is a buffer. Hemoglobin in red blood cells acts as a buffer, helping to regulate pH in the blood by binding to protons H, especially during co-transport from tissues to the lungs. pH and disease. Changes in the pH of biological fluids can indicate or contribute to several health conditions. Acidosis, a condition in which the blood pH drops below 7.35. It can occur due to respiratory acidosis, increased CO, or metabolic acidosis, increased acid production or decreased bicarbonate. Alkalosis, a condition in which the blood pH rises above 7.45. It can occur due to respiratory alkalosis, decreased CO levels, or metabolic alkalosis, excessive loss of acid. Previous year questions PYQs with answers 1. CSIR Net Deck 2017. Which of the following is the most important buffer system in human blood? A. Phosphate buffer system. B. Bicarbonate buffer system. C. Protein buffer system. D. Ammonium buffer system. Answer. B. Bicarbonate buffer system. The bicarbonate buffer system helps maintain the pH of the blood within the physiological range of 7.35 to 7.45. 2. CSIR Net June 2019. What is the role of the Henderson-Hasselbalch equation in biological systems? A. It calculates the pH of weak acid solutions. B. It describes the relationship between pH and the concentration of buffer components. C. It measures the pH of protein solutions. D. It helps in understanding protein folding. Answer. B. It describes the relationship between pH and the concentration of buffer components. The Henderson-Hasselbalch equation helps predict the pH of a buffer solution by relating it to the concentrations of acid and its conjugate base. 3. CSIR Net Deck 2020. Which of the following is true for the phosphate buffer system in humans? A. It is the primary buffer in the blood. B. It operates predominantly in the kidney and intracellular fluid. C. It maintains the pH of the stomach. D. It operates only in the digestive tract. Answer. B. It operates predominantly in the kidney and intracellular fluid. The phosphate buffer system is most effective inside cells in the kidneys, where phosphate concentrations are high. This concludes the lecture on pH, buffers, and biological significance. Let me know when you're ready for the next topic or need further clarification. You said, buffer, ChatGPT said. Here is your CSIR Net JRF Life Sciences high-quality lecture on Unit, Buffers in Biological Systems Introduction to Buffers. A buffer is a solution that resists changes in pH when small amounts of an acid or a base are added. Buffers are critical in maintaining the pH stability of biological systems, ensuring that enzymatic reactions and cellular processes occur within optimal pH ranges. The buffer capacity is determined by the amount of acid or base the buffer can absorb without causing a significant change in pH. Buffers are composed of weak acids and their conjugate base or weak bases and their conjugate acid. These buffer systems are especially important in maintaining the proper pH of intracellular environments, extracellular fluids, and blood. The mechanism of buffers. A buffer works based on the equilibrium between an acid and its conjugate base. The buffer can neutralize added acids or bases due to this equilibrium. When an acid H is added to a buffer, the conjugate base of the buffer will accept the proton H, forming more of the weak acid form, thereby minimizing the increase in hydrogen ion concentration and thus preventing a drastic drop in pH. When a base O is added to a buffer, the weak acid will donate a proton H to neutralize the hydroxide ions O, forming more of the conjugate base, thus preventing a significant increase in pH. Henderson-Hasselbalch equation. The Henderson-Hasselbalch equation is used to relate the pH of a buffer solution to the pKa, acid dissociation constant.